everybody, to this ISAPP webinar titled Human Milk Oligosaccharides, Prebiotics in a Class of Their Own. My name is Mary Ellen Sanders, and I serve as the Executive Science Officer of the International Scientific Association for Probiotics and Prebiotics. ISAPP is a nonprofit organization dedicated to advancing the science of probiotics, prebiotics, and related substances. We are so fortunate today to have our speaker, Dr. Ardythe Morrow, Professor and Director of the Division of Epidemiology in the Department of Environmental and Public Health Sciences at the University of Cincinnati College of Medicine. Professor Morrow has several patents on human milk oligosaccharides as novel therapeutics for a number of conditions, and is actively investigating the prebiotic function of HMOs as well as their overall health effects. I would also like to introduce our co-hosts for this webinar, Dr. Karen Scott from the Rowett Institute, University of Aberdeen, and Dr. Marguerite Schotterman from Friesland Campina. Marguerite, can you share the goal of this webinar?
Yes, this webinar aims to inform about human milk oligosaccharides, what they are, their prebiotic potential, and how they can impact infant and adult health.
Thank you, Marguerite. And Karen, why is this topic important?
It's really important because these HMOs are the key growth substrates for many of the first bacteria colonizing the infant gut, particularly bifidobacteria. And the variety within the HMOs impacts on the variety of bifidobacteria present. Because bifidobacteria generate really a range of diverse metabolic products in the gut that contribute to the baby's health, it's really important to boost their numbers.
Great, thank you, Karen. So now it's my great pleasure to welcome Professor Ardythe Morrow. Ardythe, can you share? Oh, your slides are shared. Thank you.
Thank you all for organizing this session. I am delighted to be speaking about one of my very favorite topics, besides my children and grandchildren, but the human milk oligosaccharide, which I've been doing research on for several decades, along with other collaborators across the world. And my message today is that human milk oligosaccharide, it has a strong prebiotic function, but it should be considered in a class of its own. And I'm going to try and advance here. Let me see. Practice that. There we go. So the themes of this webinar are really the concept of human milk oligosaccharides as prebiotics, but also their distinction and why I say that they're in a class of their own. Review them as a multifunctional food of infancy, but also then to address their potential at later ages for potentially inflammatory bowel disease or various other conditions that affect individuals worldwide at later ages. And then a brief consideration of our learning curve, which we still have very substantially, and what we need to know in order to achieve effective translation for individuals at all ages. So first, let's address human milk oligosaccharides, or HMOs, as prebiotics. So from the ISAPP website, which I think is a very definitive definition of what a prebiotic is a substrate that is selectively utilized by host microorganisms conferring a health benefit. And so typically prebiotics are carbohydrates or types of soluble fibers that humans cannot digest, but instead serve as a food for beneficial microbes. And this little schema is again from the ISAPP website and some of its available materials. And you see the prebiotic there in a heap, perhaps like a teaspoon full of sugar that we would refer to as a prebiotic here. And its mediators for health effects are really the effects of microbiome modulation, composition and function, and then the metabolites that those microbes produce, short chain fatty acids, including butyrate propionate acetate. So then what are human milk oligosaccharides? Well, they are well known worldwide as carbohydrates of human milk that are 3 to 32 sugars in length, individual monomers, that is, that are based on lactose, which makes them distinctive, to make more complex structures. And they are indigestible to humans and selectively utilized by host microorganisms, thereby conferring a health benefit. So yes, they are definitely prebiotics. More on the human milk oligosaccharide is that they are, in human milk, the third most abundant component or fraction. And following lactose and lipid, this little vial is from my own freezers, just overlay here, that HMOs at 5 to 12 grams per liter, depending on stage of lactation and which lab is measuring, but are similar in concentration than to protein. Give that some thought, that for a child that is in growth mode, that HMOs would be as or more abundant than protein in human milk. Overall, there's not just one molecule, but there could be up to 200 or more individual HMOs molecules. But typically when we're measuring those in any significant abundance, there are more like 12 to 20 individual HMOs that are commonly found in mother's milk. 70% are neutral and about 30% are acidic, and that also varies with stage of lactation. The amounts, the exact amounts are not constant, either between mothers or possibly even between populations, but they're affected by some characteristics that I'll be talking a little bit about. And to give you more of a concept of what the HMOs are made of, I'm showing you here what I call the galaxy. Now, remember the word galaxy, it really originates from a Greek word and is related to the word galactose. Now, and I'm going to show you that that's important. Galactose, represented in the cartoon here as a yellow circle, is part of the disaccharide lactose. The other blue circle there is glucose, and together they make lactose. The other parts of the cartoon here are the other 3 monomers, sugars that form the various types of human milk oligosaccharide. And here are some of the precursor molecules made by lactose. You'll notice that all of them have this galactose-glucose combination and a beta-1,4 linkage and somewhere in the molecule. And then distinctive to human milk, again, is the idea of having a fair amount of molecules that add the sugar fucose in various different lengths of structure, some of which become much, much longer than these. And then some which add sialic acid, which is that little purple diamond or pink diamond in some of my slides, again in varying lengths. So this is the HMOs gallery and just a top set of the most, some of the most abundant of the oligosaccharides. HMOs structurally are similar, and this is one very unique feature relative to any other prebiotic type. It's structurally similar to gut oligosaccharides or carbohydrates that decorate the surface of the GI tract and are abundant in the mucin layer. So on the right in milk, I'm just showing one of the many oligosaccharides called 2-fucosyllactose, one I've done the most personal research with and on. But you see then on the left-hand side, a homolog structure that would be found in the GI tract, in which in the literature in infectious disease and gastroenterology is often referred to as H antigen. This is also a structure found in saliva. Another feature of human milk oligosaccharides is that some of them are affected by genetic polymorphisms. Famously, there is a major polymorphism known in the gene that affects the linkage of fucose in an alpha-1,2 linkage to this backbone that requires an active fucosyltransferase 2 or secretor gene allele. And about 75% of the world can make this structure that you see up here on the right and on the left, both in milk or in the gut or in the lung or in saliva. And about 1/4 of the world cannot, based on their polymorphism. And there are a few other polymorphisms that affect things. There's certainly promoter regions that can be affected. But for the sialyl oligosaccharides, there is no major or any polymorphism that affects the healthy individuals. In fact, that seems to be a fatal kind of polymorphism in genes. But for the fucosyltransferase gene, this affects milk composition. And I'll show you here how. And this is a listing on the left here of every color here is representing different type of oligosaccharide. And on the left bar, what you see is a secretor mother's milk composition represented. And on the right bar, a non-secretor mother's milk composition. About 3/4 are secretors, again, about 1/4 that are non-secretor. And the first thing you can say, well, there's a lot more colors and there are a lot more bars or sections in the secretor. And that is representing that, in fact, secretor mother's milk has more diversity of oligosaccharide. There are a set of oligosaccharides that would be missing then in the non-secretor mother's milk. And this could affect the total milk composition. Also, typically, but not always, that non-secretor mothers may have as much as 1/3 less oligosaccharide in their milk. And this does seem to be dependent on stage of lactation and some other factors. Other oligosaccharides will be found more abundantly in non-secretor mother's milk, such as the acidic typically, or 3-FL, which is a sister molecule to the ones made by secretors. So there is definitely compensation. There is no such thing as inferior mother's milk. So another key point is that human milk oligosaccharides differ in structure from other prebiotics that are out there and available in the marketplace. So again, just representing one of the many structures of human milk here, also all made from these 5 monomers. And look then at fructo oligosaccharide, or FOS, and this is the combination then of fructose molecules in a chain, and at the end of that chain, sucrose, which is a combination of fructose and glucose, sorry. Then there is very abundant and very much out there and similar, and more similar in some ways, is galacto-oligosaccharides, or GOS. Or if I were Dutch, I would say Haas. This is one of the things I learned by working with Fries and Campina and my Dutch colleagues. And that is these 2 colors there, galactose and glucose, essentially made of lactose, but in longer chains and in combinations. And there's not just one Haas or GOS structure, but multiple different kinds of permutations. But I've been now talking about structure. Structure, however, follows function. And so there are some functional differences between human milk oligosaccharides and other prebiotics. So again, prebiotic functions would be those where it's occurring as a result of the utilization by microbes, by definition. But HMOs are multifunctional agents, and they have also some direct functions, direct to tissue or direct to cell in various locations that are not due primarily to utilization by microbes. And I would like to point that out as I go through, and this is an important theme of the talk overall. And in the general multifunctionality of HMOs, I really want to say that there is a fundamental triad of connection that explains a lot of the power and effect of HMOs. And it's represented in this schema. That is, okay, incoming, it's dietary, it goes into the gut, but from the gut, and what happens there with microbiota can have an important effect on brain development for the child, certainly on immune system for the child. And this triad is an important interaction. Represented here are gut cells and immune cells and the vagal nerve between the gut and the brain. So we'll be referring to that in different following slides. And this gut immune brain connection also holds for other kinds of prebiotics. It's just for the HMOs, it may have specific pathways. So now I'm going to talk about HMOs as a primary multifunctional food of infancy. And again, in this triad, I'm going to focus on its prebiotic impact in the gut to create a healthy gut microbiota, and optimizing host defense, and a little bit about enhancing neurodevelopment, which is really one of the more recent domains of research. And it's exciting and promising, and there's a lot going on there, but we'll just be covering it a little bit. So in terms of the prebiotic effect of HMOs, it's long been known that human milk-fed infants are colonized with more bifidobacteria, and that formula-fed infants typically have far more abundant what is now called Clostridioides or C. difficile. And that this is a distinction that is seen again and again and again. The key for infant colonization, as Dr. Scott had mentioned already, is bifidobacteria. This distinguishes in many large studies that have been done that the bifidobacteria are significantly more abundant in breastfed children. And it's also been shown that bifidobacteria typically, not always, but typically grow well on HMOs. There are many different species and subspecies of bifidobacteria, and they really have different capabilities. Some grow better in adulthood with different diets, but as a genus, generally, bifidobacteria typically are growing on HMOs. The champion grower among the bifidobacteria on HMOs is Bifidobacteria longum subspecies infantis, or B. infantis for short, but the other ones also do. In fact, then research has shown that organisms that are typically fermenters, Bacteroides, Parabacteroides, Roseburia inulorans, and other organisms that are fermenters can also grow on HMOs, although often will be outcompeted by the bifidobacteria when in the infant gut. And Clostridia and gram-negative organisms, which are pathogens, do not grow on HMOs, although they can grow on pieces of HMOs when they're taken apart by other organisms. So to look at the HMO's prebiotic effect here, I want to use this slide that I have borrowed from others and show that basically with the incoming oligosaccharide, thing we may focus on is that they are going into the gut, producing short chain fatty acids, and then those short chain fatty acids feed the gut wall and can also be absorbed. But there's also more direct effects. Again, this back to the prebiotic plus, is that, for example, 2FL and some other human milk oligosaccharides have been shown to be able to interact with the epithelia, reduce inflammation, and can change cell surface carbohydrate profiles. So there are both prebiotic and direct kinds of effects. I'd like to share a few studies that I had worked on with colleagues here in Cincinnati, and then some other studies published elsewhere, again, showing that there is really some profound possibilities, and I'm really focusing in on animal models, as we do not have a lot of human trial data. In this case, we fed the molecule 2FL, which again is typically the most abundant of the human milk oligosaccharides. And we had reason to think that it might benefit the gut children with who have to undergo a very severe gut surgery called ileocecal resection. Ileocecal resection is done in children after they have this devastating disease called necrotizing enterocolitis, a disease of preterm infants. And that ends up with a much shortened gut. So what they need is to be able to restore the gut architecture, be able to get back into growth mode, and to have a lower inflammation and to help the child grow in general. So in our mouse model of ICR, which can only be done by a few very talented surgeons, you can imagine a tiny little mouse and having to do a little gut surgery on them. This was done by my colleague at Children's, that we showed after a result of this experiment involving 12 mice, all of whom had ICR, that half of them were given 2FL as part of their post-surgical treatment. And it found about a 10% increase in growth above their normal chow. It restored the gut architecture of the gut. It increased the beneficial microbes of the gut, specifically in this case, varieties, which is found in the crypts, and increased the bioenergetics of gut cells. Now, I'm particularly enthusiastic about this kind of work. This was using the biopsy from the mice in the colon and looking at the transcriptome. And what we saw is about 200 or more genes significantly upregulated at fourfold or greater when they were exposed to 2FL, human milk oligosaccharide. What we saw was evidence of mitochondrial involvement, energy processing, ATP synthesis, kinds of gene expression, and also biosynthesis of sterols, cholesterol, and nucleosides, and genes involving host microbial interaction. I think this is very exciting and speaks to some other needs in other conditions that I will come back to in a few slides. But besides that function that is largely prebiotic, because that work is an example of a powerful prebiotic effect predominantly, there is another function which was first described many decades ago, perhaps in the 1950s, that human milk oligosaccharides could be binding to specific pathogens and inhibiting infection. The group that I was with for many years, NIH funded, was specifically on that topic, looking at pathogens that might particularly bind to the fucose and this alpha-1,2 linkage on the gut surface. And it's been shown by CDC and others that secretors are at increased risk of many kinds of noroviruses, rotaviruses, also Campylobacter, diarrheagenic E. coli, Vibrio cholera, and H. pylori, and that diarrheal illness, even in European populations, is significantly increased in secretor individuals, under 5 at least, through GWAS studies. So here's this cartoon: incoming pathogen has an adhesin, binds to the H antigen receptor, which is kind of shown here in cartoon. But when human milk is available, or the human milk oligosaccharide that is structurally similar, it can serve as a receptor decoy. And this has been shown multiple times in vitro studies. And for some pathogens, for example, some of the influenza viruses, or even potentially COVID, there may be a predilection for the sialic acid structure. So this is not unique solely to the fucose secretor positive structures. It can be a pathogen receptor interaction that human milk can interrupt. Here is showing as a mouse model published by my colleague Guillermo Ruiz-Palacios, showing the concept in which he challenged 2-week-old bulb C mice with 10 to the 4th CFUs of invasive Campylobacter strain. and then gave half of them an oral 2FL by gavage. And in this one image here, what you see is that in the group treated with 2FL, the carriage of this invasive Campylobacter strain was significantly reduced by about half compared to control. So it's not an absolute loss, but it is a significant reduction that could affect severity or duration of illness. Now, going beyond the microbiota now into perhaps effects that are related to the microbiota, but affect how the child or the individual who's affected by a pathogen might respond. And in this case, it was an experiment done by my colleague David Newberg in his lab, showing if the treatment of gut tissue with 2FL following exposure of that gut tissue to a pathogenic E. coli. It was aggregative and invasive E. coli. And what we used as a readout was TNF-alpha on gut tissue, which is a very well-established marker of inflammation. You can see from baseline low TNF-alpha exposure to the pathogenic E. coli significantly increased TNF-alpha. And then when the gut tissue was treated with 2FL, the TNF-alpha inflammatory marker decreased to the baseline level. But 2FL is not the only human milk oligosaccharide that can affect tissue directly. This is a lovely review article that I encourage you to look at, in which this is looking at a variety of different immune cells and lectins that's been as a review from many different publications, and how human milk oligosaccharides of various kinds can bind receptors on immune cells and affect their function. And this is a very specific structure-function relationship, kind of like lock and key. Only certain HMOs affect certain types of, bind with certain lectins on these immune cells. And you can see here in one of the columns here, the known HMO ligands. So then finally, I'd like to just give a very quick review of how HMOs can affect neurodevelopment. Again, this is one of the more exciting, I think, and recent developments. The idea that, nutrition is just what you do in the gut. I think we've sailed way past that concept to recognize that it has systemic effects, immune effects, but also very much so neurodevelopment. And this is so important for the growing infant, that growing the brain and its capacity is one of the things that is most important to parents and to the child over the course of their lifetime. The ways in which human milk oligosaccharide and potentially other prebiotics could affect neurodevelopment have to do with the bacterial secondary metabolites, metabolic precursors, certainly immune signaling, including whether the gut is inflamed or not, or their inflammatory markers. The vagal nerve is a really powerful connection between the gut and the central nervous system, and its signaling can have important effects in the brain. And HPA axis activation. These are all potential kinds of mechanisms. And what has been well established is that in germ-free mice, or in individuals who are depleted of gut bacteria by antibiotics, a phenomenon that's all too common in infancy. And in Cincinnati, by the way, we have shown that maybe 60 to 70% of mothers have antibiotics at the time of, around the time of delivery or just before delivery. So this is a very concerning issue for me. And that we need to understand this. My argument is that we need to really understand what's happening to the normal physiologic process of the colonization of the gut in order to know how we can intercede to provide the kind of messages to the brain and its development that are needed. So the HMOs do not enter the brain directly. For a while, it was thought that perhaps that was occurring, but that has been shown not to be the case. But it has been shown that through the growth of the microbes, that is the prebiotic effect, that HMOs via microbes stimulate the vagal nerve, can produce neurotransmitters or their precursors, and modify immune signaling, and thereby affect brain function and neurodevelopment. And that sialic acid from oligosaccharides is the one domain where a component of the oligosaccharide that is specifically sialic acid itself might contribute directly to incorporation into the brain. And here are just a couple of nice papers. One by Vasquez et al. In 2016 showing through an experiment in operant conditioning and mice fed the 2FL, particularly that they could shift the potentiation of the gut-brain communication. It was clearly via the vagal nerve. And then by Mudd et al., looking at dietary sialolactose influencing the sialic acid content in the prefrontal cortex, and by MRI imaging, looking at the corpus callosum of young pigs. So these are both, of course, animal models. Well, then here we give this broad, quick overview really of human milk. And there's so many potentials there that we see for gut health, for brain health, for immune health, potentially as prebiotics, as pathogen binders to an extent, and many different functionalities. But these are all in vitro or in vivo, in animal models. So what do we know directly about trials in human infants? I'm an epidemiologist, and I am not showing some of those data today, but there are studies that are epidemiologic that indicate important health effects. In 2004, I published data showing reduced diarrheal illness and its severity associated with having more 2FL in mother's milk. That's the kind of thing you can show in an epidemiologic study. But what I'd like to focus on for the few slides are some of the few trials that we have. And one of the first of the randomized controlled trials that I'm aware of was shown here is using the combination of 2FL and GOS in a trial of 420 infants. Major focus was to look at tolerance and growth, as well as immune modulation. The total amount of oligosaccharide was kept the same. The control formula had 2FL grams per liter of GOS. There are two treatment groups, one where it had more 2FL with 1 gram per liter and balanced with the rest as GOS, and the other one a smaller amount of 2FL, only 0.2 grams per liter, and the rest GOS. And then all of these trials typically have an exclusively breastfed comparison group. So in this trial, growth was the same, tolerance was clear, but I was particularly intrigued by, again, looking at the readout in the peripheral blood on TNF-alpha levels, and measured here on you can see on the y-axis, the control formula had highest circulating TNF-alpha. And the two formulas that included some amount of 2FL had significantly lower levels of TNF-alpha. That gold bar across is showing you what the level of TNF-alpha is in human milk, the exclusively breastfed babies. So this is showing that when adding this oligosaccharide, you can get TNF-alpha effects within the same range as you would find in a breastfed child. A second randomized controlled trial published a few years later, 175 infants, had a different kind of formulation of 1 gram per liter of 2FL combined with its precursor human milk oligosaccharide called LN-NEO-T or LNNT. And that was at 0.5 grams per liter. So together is 1.5 grams per liter. And comparing to control infants, there were some really significant health effects that lowered bronchitis, lower respiratory tract infection, and lesser antibiotic use. I found quite intriguing. Entirely different set of readouts from the first trial. And then finally, one of the more recent trials, several of the recent trials have been looking at human milk oligosaccharide combinations, using some of the more abundant and representing some of the different types in that galaxy of HMOs, some of the different structural themes. 2FL, fucose, LNT being a precursor here, 3SL and 6SL containing sialic acid, and the DFLT was another fucose containing molecule slightly longer. And this trial, published by Bashiva just this year, looking at the impact of this combination on the microbiome. And the 2 test groups they had was one with 1.5 grams per liter of this combination of oligosaccharides, the other test group having 2.5 grams per liter of this combination, and then the control formulation just being a standard formula without oligosaccharide. So this is an important paper because it's also showing that at that level of oligosaccharide and this diversity of oligosaccharide, able to show an increase in bifidobacteria, relative abundance by at least 3 months of age and on to 6 months of age, comparing the test groups to the control, and closer to human milk, which is that 1 bar HMG, the way they have abbreviated it here. So more bifidobacteria, and another important readout is less C. difficile. So another marker for formula feeding. And you can see at baseline, there was no difference in these groups, but by 3 months of age, the control formula had significantly more C. diff at 3 and 6 months compared to any of the test formulations, or of course, the human milk fed infant. So again, this is promising for the shift in prebiotic effect towards more like human milk. Now I'd like to, in a few remaining minutes here, address the health needs of adults and people at older ages that might be relevant, or for which HMOs might be relevant and could be thought of as something by using synthetic human milk oligosaccharides to provide to people who are not going to be breastfed directly. So an interesting paper in 2002 in the New England Journal of Medicine showed on the left-hand graph that since the 1950s, and it's really from before that, until about 2000, and a vast decrease in infectious disease. Now, post-pandemic, we may not be so confident that infectious diseases have really been conquered or anything. We've been chastened. We know that we are still susceptible to infectious disease. But we also have this other ongoing issue of a rise in autoimmune and allergic disease, which is graph B. And in the literature over time, since the revolution in microbiome analysis by the mid-2000s, about 2004 2014 or so, we had a rapid increase in publications on dysbiosis, meaning gut dysregulation, microbiome dysregulation, and disease outcomes. And that's because dietary issues, shifting microbial exposures, heavy use of antibiotics globally, and C-section delivery at least, and other environmental exposures that are new to our environment, to our modern life. So could HMOs be helpful at older ages beyond infancy? Well, here's to test even whether there was tolerance and impact, a prebiotic impact. There was a study done by Ellison et al., randomized controlled trial on 100 healthy adult volunteers consuming several different oligosaccharides. One was 2FL, and I think the other one was LNUOT for 2 weeks. And it was found to be safe and well tolerated, and that at 10 grams per day effective dose could shift microbial colonization towards increased bifidobacteria and lowered potentially pathogenic bacterial profile. In some conditions like inflammatory bowel disease, we know that the microbiota are an ongoing problem and need some solution. And also in that maybe when I looked at this profile for IBD in particular, I saw lots of possibilities. First of all, we see increased gut inflammation in IBD. We've seen that 2FL can address and reduce inflammation. Again, the lack of beneficial microbes, increased pathogens in IBD. Could 2FL provide some benefit? Potentially, yes. That there's a problematic gut mucosal damage and altered architecture. Studies I talked to you about suggest this 2FL could be helpful. And a suppressed gut mitochondrial energy signature that we see in IBD, we could actually potentially be rescued in part by 2FL or other oligosaccharides. And this is just, again, connecting two different worlds of research. The paper I showed you by Mazoff et al. Showing this energy signature in the mice treated after surgery, but also publications and research on patients with IBD seeing a very similar profile of suggesting the need to energize the gut microbiota through a fermentation and prebiotic effect that HMOs might help address. So we are doing a trial that is my colleague, Dr. Ted Denson. It got delayed by the pandemic in part. It's been sort of hampered, but it's now back up and running, looking at tolerance first in IBD patients, what dose level we should use, how it's affecting the microbiota, looking for a decrease in pathogens and an increase in beneficial bacteria, reduction of inflammation. And first, the adult study is required by FDA. That's what we're doing. And then a child study, because children as young as 3 and even younger are getting IBD now. So there are other possible applications. There's another trial going on in Cincinnati for bone marrow transplant to reduce gut inflammation and sepsis. Irritable bowel syndrome has been another proposed use. The potential for immune modulation as an adjuvant for vaccines, supplement to reduce risk or severity at older ages, autism, and allergy. So there's a lot of applications, but as we talked about, there's important multifunctionality, and it's about matching up the specific oligosaccharides and the specific needs. So we are still learning a lot. I like this little quote from Michelangelo. I'm still learning. That's certainly me and the whole field of HMOs. And so what we need is a big learning curve in which, for the infant feeding side, what are specific HMOs doing? What combinations of HMOs are really optimal? We're never going to replicate human milk exactly as we have it. It's also quite varied. So how many HMOs of what structure? So this is where the field is working. And what outcome differences can we obtain through trials? And then at Health at All Ages is really mapping structure function to the issues of those health conditions, and increasing both the basic science and the trials that are done in any given domain. So with that very rapid, broad overview, I thank you for your attention, and I'm delighted to field any questions.
Thank you very much, Ardythe, for that extremely interesting and informative overview of HMOs. And I can see that we have a lot of questions coming in already, so we will now open up the floor to questions. So Marguerite and I will be chairing the question session, and we'll pick questions out of the typed in Q&As. And I will just start off, if that's okay. Yes. So the very first question, I think, is actually a very good overview question, in fact, from Marcus Glitvitsky. Sorry, apologies if I pronounced that wrong. Do you believe that long-term HMOs will completely substitute GOS and FOS in infant formula?
I have no evidence that that's going to be the case, but I would say the more infant formula can be closer to what is found in mother's milk, all of the infant formula efforts over time have been trying to get it closer and closer and closer. There is the issue of cost and function, and we want to be able to demonstrate there's value for what we're doing. Note that GOS is structurally more similar, so that it's possible that it can also be part of the mix in formulas, but we'll see. And so what I hope for actually is a mix of oligosaccharides where we know what their functions are contributing together, and then seeing whether that also needs to be augmented with anything else.
Yeah. Yeah, so I mean, what you're saying is that the simple fix at the moment has been to put GOS in formula milk, and right. we're getting better at realizing that that's not enough on its own, perhaps.
Right, exactly. No, I think that's right. It's very important that babies get some form of prebiotic, just like we should at every age, if we do the best we can and we keep moving it along. That's what I'm hoping for. And so the question is, eventually, I think so.
Okay, that's great. Thank you. Marguerite?
Yeah, thank you, Ardythe. Another interesting question which came in. Does the mother's diet have an impact on the levels of human milk oligosaccharides in breast milk? And do levels of human milk oligosaccharides in the breast milk have an impact on gut microbiome profile of the infants?
So I'd like to show a slide that I kept behind, if I can for a moment, but maybe I can't now. I'm not sharing. That's all right. We need evidence in this direction. There is evidence from a probiotic trial that was done in Finland that if mothers who were given an experimental basis probiotic or just a placebo, that the human milk oligosaccharide profile differed somewhat. So it is evidence that if mother's own colonization is somewhat different, they could be more capable of making one or another of the human milk oligosaccharides. So that was a probiotic trial, but I think it's possible with the prebiotic to have some effects as well, and we need better studies and in that direction. But it's a possibility.
Okay, thank you very much. Karen?
Okay, so what I think one interesting question that's been asked, Ardythe, is are HMO effects on the infant's brain and immune system transferable to adults? And is there data for this?
So these mice models that I showed you really are not necessarily models of infancy all the time. Some of them are, and there is evidence in general that some form of prebiotics can in fact influence the brain. And the question is whether it's neurodevelopmental or whether once the brain is largely developed, if there can be effects. So some things that are at older ages might have to do with, for example, inflammation in the gut as a negative influence, having things like brain fog or anxiety, depression. There is evidence from some of the work that's published and some things that are in progress and not yet published, suggesting that oligosaccharides can help with some of the effects like anxiety, depression, and I think that there's real possibility. Some of the effects, of course, are very early neurodevelopmental effects. So we'll have to separate those, but I think we've got possibilities at various ages.
Okay, thank you.
Okay, another question. Are there any data indicating benefits of human milk oligosaccharide supplementation to healthy infants and young children following completion of normal breastfeeding? So for instance, after 6 months.
So the trials as required in the by FDA in the United States, and I imagine the European Union may be similar, typically want trials where it's very clean and that you're just giving one thing or the other and you're not switching over. But the animal data and the general functionalities suggest that this could be very important. And I, for one, would love to see trials that are mimicking what is the real practices of mothers who are breastfeeding, which is that a lot of times moms struggle. They work really hard to breastfeed for a period of time. Then they need to switch over to formula. And when they do, they would like to have the very best formula. And so my best guess is, based on the evidence, all the evidence we have in the lab and elsewhere, that it would. But the trials are not going to at first be informing us in that direction. We would have to infer that it would be, but I think that would be a great guess to make.
Okay, thank you.
Okay,
Karen?
there's two quite linked questions, so I'm going to ask them sort of as a double question, actually. So somebody's asking if you think that 2-FL is the best HMO, and then there's another question wondering that given the complexity, and they keep flickering away the question, sorry, I'm trying to find it again. A question about producing HMOs, given the complexity of HMOs that exists, how can we, is there a really easy way that we can synthesize them? Or why are there not more manufactured HMOs already? Because we've known for a while that there are more than 200 different HMOs in human breast milk. So yeah, that's right. Why has there not been more developed?
Yeah, these are great questions. So 2-FL, is it the best? Well, there is no one best, but I would say that the field did start more focused on 2-FL. Why? Because it's the most abundant of the human milk oligosaccharides. There was in the group that, for example, I was working with, NIH funded thematic representation of that structure in regard to risk of diarrheal diseases, where we happen to be working. Lots of work has been done on 2-FL, more than other oligosaccharides, but a lot of other oligosaccharides are also being looked at, and it takes time. This is a structure function thing, and we've got to drill down to really understand. So science just takes its own time. Is it, I love the molecule 2-FL, but I wouldn't say that it's really the best. I would just say it's one of the important oligosaccharides of human milk. In regard to the question about producing this array, this galaxy of human milk oligosaccharides, this is also a substantial challenge. For a while, it was so difficult that it was impeding our ability to even do animal studies. So that it was for a decade or more, all we could really do was very few animal studies, very small, and really do in vitro work. So now there's been breakthroughs in the bioengineering of E. coli and other perhaps other organisms too, but the E. coli that it can produce synthetically. We tried a lot of different methodologies. You can go back in the literature for about 5-10 years, and you would see people trying to do transgenic transgenic goats or cows, and that was not going to do it at all. So now it's and bioengineering organisms that can make it just the way they make vitamins, you know, same methodologies here. And being able to make these very specific structures in just the right conformation, and then being able to afford. So now we are in a range where we actually have a set of human milk oligosaccharides to work with. That's been an exciting breakthrough and now enabling, but getting to 200. So typically moms are not making 200 that we can really measure in any appreciable amount. The appreciable amounts were more like 15 at most or 20 at most. And they have some thematic similarities. They are, the less abundant they are, the longer they are typically, but there's the same fucosyllactose acid, glucose, galactose, N-acetylglucosamine. You take these 5 monomers and it's like tinker toys. You can put them together in different ways, but we want to make them to be structurally identical to what's in mother's milk, if at all possible, so that we can call them human milk oligosaccharides, and we can map their functions to functions that are happening in the breastfed child. So I think the goal now is to get to an array that have these different thematic structures that are mapped to functions that we need, and then to remain affordable to do and in the near future. That makes sense.
Yep, that's great. Thank you.
Okay. Is there still time for a question? I'm also looking at Mary Ellen.
You got 2 minutes.
Okay.
Yeah, go ahead. I think we have time for one more.
Okay, for the non-secretor type, is it a genetic defect or is it meaningful to some extent? And we say that breast milk is the best nutrient. Is mother's breast milk the best for her own baby? And even if it can't secrete 2-fucosyllactose, does it really make sense we fortify 2-fucosyllactose in infant formula products for all the babies?
Mm-hmm. So lots of questions in that one question. So first of all, worldwide and for countless millennia, there have been secretor moms breastfeeding non-secretor babies and secretor babies because any secretor mother might also have a non-secretor baby. It's just, you know, it's Mendelian genetics. So this has been going on forever and a day, and there are benefits that, and there is no, it's not like ABO transfusions or something like that. This is a universal donor situation. You can give all of the above, as long as it's coming structures in mother's milk. So there is no inferior milk. There's never been that evidence, but it depends on you know, just like, okay, I'm really tall. That's good for getting dishes off the top shelf, but I'm really lousy at getting dishes if they're too low in the kitchen. I don't want to get down there to get them. I'd prefer a short person do that. That's how I think about the variations in everything that we have genetically, and that basically there are benefits and perhaps somewhat not a generic deficit, but things that something's better at or, you know, less at, but overall equivalent. So I think that's part of our learning curve right now, about to understand how that really functions. But no mother should worry about whether she's secretor or non-secretor. And also there should be no worry about putting 2FL in whatever formulas, because that's been going on in nature for a long time.
Okay, thank you very much.
Yeah, unfortunately, I think our time is up now. What a great discussion. We have quite a few questions we were unfortunately not able to get to, but I want to thank Ardythe so much for what a great talk and a great topic for everybody. And the audience, thank you so much for so many great questions and appreciate your participation. And Marguerite and Karen, thank you for hosting. You did a great job. And just to remind everyone, a recording of the webinar, and this will be fully open for anyone to view, will be available on the virtual event content section of the ISAPP website within a few days. We'll send a follow-up email that has a link to that section of the ISAPP website. So look there for the recording. So thanks so much, everybody. Bye-bye.